Hello, my name is Sheila Kitchen. My pronouns are she, her, hers, and I'm currently a postdoc in the Parker Lab at Caltech. My talk today is on the genomic and biosynthetic evolution of a key chemical innovation in rove beetles. Rove beetles belong to the family Staphylinidae, a megadiverse group of 64,000 predominantly predatory species. The diversity of this group is attributed to their short elytra, or wing case, which has freed up the range of motion of the abdomen, allowing them for more efficient movement in the soil and leaf litter and expansion into new habitats. The species richness, though, is uneven across the subfamilies within Staphylinidae. The largest family, or subfamily, being the Aleocharinae, a clade of nearly 17,000 species. The radiation of this group is attributed to the second key innovation, the defensive tergal gland. This structure, positioned in the flexible abdomen, produces a potent chemical secretion that is effective for evading predators, as seen here by the beetle escaping the ant. Consistent with the tergal gland being a key innovation, the early branching lineages of aleocharines and outgroup lineages that lack the gland are species poor. The gland is also proposed to allow rove beetles to infiltrate ant and termite colonies, leading to many social symbiotic relationships across the clade. In several of these specialized symbiotic taxa, the chemical defense has been lost, but there are no cases of it ever being regained. To gain a functional and evolutionary view of the gland, we perform single-cell RNA-seq on the abdominal segments of Dalotia coriaria, an emerging model system for interspecies interactions. The cell type atlas allowed us to identify biosynthetic targets for functional tests using RNAi, with the GCMS chemical readout as our phenotype, as shown here by the wild type. For example, master fatty acid synthase, which is highly expressed in the solvent cells, shown in purple, and when knocked down during development, leads to the loss of all of the solvents. Similarly, we identified a terminal gene in the synthesis of benzoquinones, a lactase we call decommissioned, because it wipes out the defensive compounds of the secretion, as shown in green. This study revealed how the two cell types that make up the gland, the solvent cells and the benzoquinone cells, or BQ cells, cooperate to produce the defensive secretion through transcriptional hybridization of several insect abdominal cell types, like the enocytes, the fat body, and the cuticle cells, as well as gene duplications and co-option of other pathways that convert aromatic amino acids into benzoquinones. Building on this work, we wanted to retrace the evolution of benzoquinone and solvent cells across the diverse clade of rove beetles. We sequenced the genomes of outgroup lineages in red, early branching aleocharines in black, and higher aleocharines in blue, using primarily Illumina sequencing. We also assembled the chromosome-level assembly of Dalotia, our model system, using a combination of Illumina, nanopore, bionano, and split pool recognition of interactions by tag extension, or sprite, sequencing, which is very similar to Hi-C. While the gene complement of the genome was highly complete, we were only able to assemble 2/3 of the genome due to 50% of the genome being composed of a single satellite. We also characterized the chemical diversity of the tergal gland secretions within the clade of higher aleocharines, where the gland first appeared approximately 120 to 150 million years ago. The classical gland secretion is composed of benzoquinones in green that dissolve the fatty acid-derived compounds in purple. We found that the BQ fatty acid cocktail was common in the majority of the higher aleocharines, with the first lineage where BQs were present, dated between 96 to 117 million years ago. Using our data sets, we wanted to know what are the genomic substrates that gave rise to the gland, what are the core solvent and BQ genes shared among the higher aleocharines, and what happens when you become a highly specialized symbiont and lose the gland altogether. I won't have time to talk about this one today due to the time constraints. Comparing the first lineage when BQs are present in the gland secretion, Aleocharia, to Dalotia, we find some overlap in their chemical compounds, supporting a chemical stasis that spans 100 million years of divergence. When we looked at the cell type-specific RNA-seq profiles of differentially expressed orthologs, we found a tight correspondence of BQ and control cell transcriptomes, but a slight divergence in the solvent cell transcriptomes of the two species. When looking at the overlap of the differentially expressed orthologs, we identified a core set of solvents and BQ genes that are conserved between the two species. Altogether, this data implies deep conservation of the BQ cells, whereas the solvent cells are more flexible in their gene repertoire. Coming back to our phylogeny, we next turn to the lineage where the gland is present, but the benzoquinones are absent, the hypocyptines. This led us to ask, are the hypocyptines a transitional group representative of the developmental steps of the cell types, or have the extant species of this lineage lost benzoquinones due to the reduced selective advantage of maintaining these defensive compounds. In looking at the compounds in the gland, they are quite different from the other higher aleocharines like Dalotia and are missing those benzoquinones. To determine if the compounds were still used in defense, we took synthetic compounds of Holobus to make mock secretions and applied them to Drosophila larvae. Survival was high in all Holobus compounds in isolation and in combination. We then asked, is Holobus solvent capable of dissolving benzoquinones and therefore acting as a defensive secretion? And the answer is yes. When we added the benzoquinones to the Holobus mixture, we saw a significant drop in fly survival, nearing the response to the Dalotia chemical secretion. To identify genes shared by the gland segment with higher aleocharines, we performed segment-specific RNA-seq instead of cell type-specific RNA-seq due to the fact that these beetles are smaller than a pinhead. Comparing the expression on the x-axis, we found that several orthologs of Dalotia were enriched in the gland segment, as shown as the green and purple circles. Using hybridization chain reaction, we confirmed decommissioned the lacase that was specific to the benzoquinone cells in Dalotia that's required for the synthesis of benzoquinone was present in the BQ cells of Holobus, but at a much lower abundance than Dalotia. This had us scratching our heads. We then did in vitro enzymatic assays of decommissioned with Dalotia and Holobus decommissioned proteins, and they both were efficient at converting hydroquinone substrates into benzoquinones. Therefore, we concluded that in the case of Holobus, it is enzymatically equipped in the right cell types, but does not produce benzoquinones. This could be due to missing substrates in the benzoquinone cells or another component upstream of decommission that we have yet to characterize that has been lost in this group. To conclude, we have demonstrated that since its origin, the gland has undergone both functional and molecular stasis over deep time as these beetles have radiated globally. But they've also maintained a versatile substrate for the emergence of biochemical novelties that facilitated the lineage specialization. Hypocyptines in this case represent a unique position within the higher aleocharines, demonstrating traits that suggest they are transitional. However, we cannot rule out secondary losses. And with that, I'd like to thank the group of the Parker Lab, as well as my co-authors and the funding sources that went into the work.